Welcome. Welcome everyone.
Everyone.
We are at the second edition of PrecisionTox Café Scientifique. Welcome everyone. We are already 14 participants. I hope you enjoy the music. Hello everyone. Welcome everyone. So we are at the second edition of PrecisionTox Café Scientifique. Still the right moment to have your warm cup of tea, your coffee or a glass of water. We have still 1 or 2 minutes if you want to take it and have it with you for this one and a half an hour. We are now almost 20. We start in 2 minutes. Okay, welcome everyone. Still participants are arriving. So I will start slowly by slowly. So welcome everyone. If you join us to attend the PrecisionTox second edition of the online Café Scientifique, you are at the right place. We have participants from Europe, participants from North America. So good morning for some and good afternoon for the others. So my name is Valentin Salamon and I work for Tox, and I will play the role of facilitator today to ensure the smooth running of the event. With the help of Jeanne Lapérouse, who is communication coordinator of PrecisionTox, and my colleague at Tox, and Agatha Ormana, who is project manager from the University of Birmingham. So we are delighted and pleased to present you this second edition of the PrecisionTox online Café Scientifique. For this edition, we will spend the next 1 hour and a half with Professor BEATE ESCHER to go through the topic of cell-based bioassay for PrecisionTox and what else can you do with them. So a quick introduction of Professor BEATE ESCHER. You are head of Department of the Cell Toxicology at the Helmholtz Centre for Environmental Research in Leipzig, in Germany, and professor at the Eberhard Karls University in Germany. Before giving the floor to a professor, just quick information to make sure that everything's went well during the next hour and a half. So as you have seen, the video is recorded. So this webinar is recorded and can access after the meeting. If you encounter any technical issues with Zoom, please feel free to write it in the chat box. Agatha, Jeanne, and I will make sure that respond to your problem or help you. If you have any question regarding the content of the lecture, we will ask you to put them in the chat or to raise your hand. And after Professor BEATE ESCHER talk, we will make sure to transpose your question to the professor. So 3 quizzes will be asked during the webinar today. So stay focused. And for the rest, I wish all a nice webinar. Professor, the floor is yours.
Thanks very much, Valentin, for the very nice introduction. And thanks for having me here today. It's a great honor to be the second one after the introductory talk by John. And so I'm not sure if I can live up to that because I start very tiny. So my idea, what I would like to do with you today is take take it from the cellular point of view. So I'm asking the big question, if cells can even serve as a sentinel for PrecisionTox. That's maybe asking a bit too much. But in this beautiful picture that we is guiding us through PrecisionTox, I hope you can see my cursor. You can really see how the cell is at the basis of the phylogenetic, phylotoxic tree. And so I hope that the cells will be somehow the glue already for the project. So I'll just introduce you what we plan on doing with our cell assays in a pretty modest way. It's a very small part of PrecisionTox, but also what else we are doing, or what else you can do with cell-based assays. By the way, is the sound, everything is good, Valentin? Can everybody hear me well?
So I hear perfectly. For the moment, we have no remark in the chat, so yes.
Okay, awesome. And so our speciality, as you have said, is that we run cell assays in high throughput screening mode. So I'll show you a little bit how that is being done, and the challenge that comes with those high throughput systems, because it's really hard to understand the dosing. So I'll talk a lot about that, because that's also something that in work package 2 we are concerned about a lot. And then the interpretation of the effect concentrations. On the one hand, of course, we want what we do on the cellular level is meant to inform the omics work later in PrecisionTox, but on its own, we can also interpret the cell assay responses. And then I go beyond of what we will be doing in PrecisionTox is how cell-based assays can be applied in mechanistic toxicology and research on adverse outcome pathways in chemical risk assessment, but also in environmental monitoring and biomonitoring. And it's all really inspired by this TOX21 toxicity testing in the 21st century, TOX21, where you can see that the perturbation on the molecular level and the dose in vitro are really set in the center of this approach of new ways of toxicity testing. So we can this is just a reminder, toxicology 101, the continuum of toxicity. So as the dose increases, so does the level of biological complexity affected. So measurement on the biomolecule and cellular level may serve as sensitive early warning signals. And at higher biological levels, you also have defense mechanisms. So in a way that leads to a lower sensitivity. Another paradigm is that any biological effect caused by a chemical must be linked to some underlying molecular or cellular effect. And that is the basis of that adverse outcome pathway concept, where we go from cell toxicity via organism to population level response. And we can also zoom then into the cellular toxicity, the so-called toxidynamics, where we have an initial molecular initiating event that's followed by a series of key events, cellular stress responses that finally lead to cytotoxicity. And this is, of course, only simplified a linear pathway. At the moment, we know that there's pretty complex networks of cellular toxicity pathways, but the whole idea is to kind of root the effects, the mechanisms of toxicity in those pathways. And we have, if you just blow that up, that's again not comprehensive, more didactic, that from toxicokinetics to the cellular toxicity pathways, we have a whole suite of bioassays that match with the different important molecular initiating event, mainly based on nuclear receptors, and the cellular stress responses we detect mainly through transcription factors. So we use a lot of reporter gene assays, and that's how they work. So you have a lot going on in the cell, and you can't really see inside. So you can maybe put a dye on, so you can color the nucleus, and you can color this and that part of the cells. You can look at the cell viability, but be interested in the molecule initiating events, for instance, binding to a nuclear receptor. So what we do is we try to pick and visualize just what this nuclear receptor, and in PrecisionTox, as an example, we will be using the glucocorticoid receptor, how this is activated. So the nuclear receptor would be glucocorticoid receptor, the toxicant or any natural ligand would bind. The complex translocates into the nucleus, binds to the response element, and then not only the natural genes are transcribed to proteins, but also reporter proteins that can easily be measured. Same principle also for the transcription factors and the adaptive stress responses. Here we focus in PrecisionTox on the antioxidant response element. We pick that out, we try to visualize that, the pathway, the example of the antioxidant response element. We have Takeup as a transducer, NRF2 as transcription factor, translocation into the nucleus, binding to the antioxidant response element. Same thing, we have all the transcription of pathway-related proteins formed, but also production and detection of a reporter gene. So how do we do this in practice? In doing it in high throughput means we're doing it in a robotic environment, but it's still modular. We still have a lot of people involved that can act and work much faster than a robot, but the robot gives us kind of this continuity that we can run plate over plate over plate. So we automatically dispense cells and incubate them for 24 hours in an incubator. Then we prepare the dosing plates. We can do that with a pipetting robot, or we can do it with an inkjet. So when we have very small quantities you want to dose, type of an inkjet printer type of thing. And then we combine with this pipette that has 96 tips, so it can be done all 96 steps at a time. So I hope a few people will just start to get big eyes and say, oh, how great is that? And it is. And so we combine the dosing plates with the cell plates, and then we have the 384 well plates that we put into the incubator after we already looked at the cell confluency just by simple imaging methods. And then after 24 hours, if the cells are not damaged by the chemical, they have nicely grown over, and so we can compare the image at the beginning to the end in the unexposed and the exposed cells and can measure the cytotoxicity. After that, we typically break up the cells and add an appropriate enzyme. So we'll be using Lucifer. We break up the cells, isolate the enzyme that the reporter has produced, and use and add substrate. So we have luciferase as a reporter enzyme, and for that we just add ATP and luciferine. Or we have beta-lactamase, and for that we add a very complex FRET fluorescence reagent that gives us a very high sensitivity. And then we do dose response assessment, both for cytotoxicity and the activation of the reporter gene. When you work with complex mixtures, you often have masking of the specific effects, and that's why cytotoxicity and activation of the receptor always have to go hand in hand. A new trend is our imaging assays and cell painting, and we're going down that pathway too, not yet for PrecisionTox. And here we work with differentiated cells. That's just an example on a neurotoxicity assay on SHSY5Y cells. They're also same procedure as I just shown you, seeded in 384 well plates. The chemicals are dosed, and after 1 hour, you can already detect the acetylcholine esterase inhibition activity in the synapses of those cells using the Elman assay, or you grow them for 24 hours. And then you see here tiny in red, you see here the neurites, and you look at the neurite length. And if that's reduced, then it's a specific effect on a specific neurotoxic effects. And we also look at cell viability. There's a challenge for this application of those high throughput screening assays, and that's really, it's so small of a volume. So one of those 384 wells in that 384 well plate, so one well is a fairly complex system, but it only contains maybe 40 microliters of medium cells, and there's a lot of loss processes going on. So you can lose your chemical to the air because you can't just make it all airproof. The cells need to breathe. Chemicals can bind to the medium components, to the plastic. They can degrade. They will be taken up in the cell. And then, of course, it depends on how they're metabolized and distributed within the cell to the target site until you can finally measure your effect or see your effect. So that's a fairly complex system. And when we only look at what the cell sees, then you have, when you put the nominal concentration on one and you look at where the chemicals are in this simple system, in this complex system with a very simple model, from low hydrophobicity, so low optimal water partition coefficient, to high hydrophobicity, high optimal water partition constant, then you see that the very hydrophilic chemicals are mainly in the aqueous phase. They're not taken up very much, but let's say from log KOW4 on, the cellular concentration is fairly constant, but it's much less than the nominal concentration. And a lot of the chemical is actually bound to medium components. That sounds pretty bad to begin with, but as we'll see, it's actually not that bad because it keeps our system, our tiny systems, very stable. Otherwise, we couldn't work in such small cell volumes. So just think about dose in toxicology. So what you're used to is the dose. So you add 1.1 milligram, for instance, of the chemical to your system. That's like in an animal experiment, you would feed an animal a certain amount. Or you have that dose, but in the cell systems, it really depends on how much medium, how many cells you have, and this just compares two different scenarios. Everything's the same, but here you have 100 microliter medium, and here you have 200 microliter medium. And so what that means is, why is that not working? You have here nominal concentration of 1 milligram per milliliter, and let's say you have 0.1 microgram per cell. And in this case, you actually have half of that nominal concentration because your volume is the same, is double, while your dose is the same. So you have to be really mindful because that would translate into a smaller amount in the cell, so the actual bioactive dose. So you have to be very mindful of the different ways how you give the dose metric. So normally we would just use the nominal concentration because it's very hard to measure concentrations in those very small systems, but it's feasible, as I will show you. You can also go for total concentration. You can relatively easily measure the freely dissolved concentration. I'm going to show you that. But what we are really interested are the cellular concentrations and the target concentration, and they are not directly accessible, only indirectly by fluorescence measurements or by modeling. Before we go even into the cell, we have to make sure that the chemicals are stable under bioassay condition. And here I wanted to introduce to you the PrecisionTox team, and most specifically Julia Huchthausen, who is our PrecisionTox PhD student, who has already developed a stability test under bioassay conditions. So whenever we suspect that a chemical is not stable, we can't do that for all the 250 chemicals, but for the ones where we have suspicion that they're not stable, we can go through this system. So what we do before we even go for a test, we look at the stability in assay medium. If that's under the assay condition is the half-time, decay half-time of the parent is more than 100 hours, we don't even worry about it. We can go straight to the bioassay. If it's not the case, it's unstable under bioassay condition, we should measure the exposure concentrations, and we can also evaluate the degradation process. We don't need to do that in every case, but I just show you how it can be done in principle. So first thing, we check for hydrolysis, and if chemicals hydrolyzed, we would go for pH-dependent analysis of hydrolysis. So in case this is hydroxide or proton catalyzed hydrolysis, if the hydrolysis does not explain the half-time, it's often the case that there's a lot of proteins in the medium, so we use bovine serum albumin as a proxy for proteins, or glutathione as a proxy for the reactivity, because that's just a peptide that's not three-dimensional, so there's no reactive sites that are buried inside. So we can check if the chemical is reactive towards proteins. We can also look at oxidation. Autooxidation is a problem for certain chemicals, and we use NBS, N-bromosuccinimide, as a proxy for oxidation, which is already a pretty strong oxidant, stronger than the bioassay condition, and we looked at photolysis. So experimentally, you can also do that in well plate format. We used here 96 well plates, and we measure the concentrations with this, what we call hedgehog. So this is a SPEMI device that has solid phase micro extraction device that has 96 tips with 96 sampling phases, C18. And so we have a little shaker, and so we can shake it and accelerate the kinetics, and then quantify the chemical and the loss of chemicals in a relative manner, because it's all first order kinetics. And so that works really well. And with this, we tested 22 chemicals, and they're not representative. They were already selected as chemicals that are prone to degradation. And we saw that quite a few were actually degraded under those bioassay conditions, which means when you only take the nominal concentration, the chemicals don't look as potent as they possibly are. In these cases, we recommend measuring the exposure concentration in the cell assay. Same principle. We haven't really done that with that hedgehog because it's really new, but with individual SPEMI fibers, and you can go in the medium with and without cells, and you can even use that to measure just partitioning constants to proteins or to lipids, and or even to cells. And so with this, we can actually also measure the effect concentration, the freely dissolved effect concentration. We call them inhibitory concentration for 10% reduction of cell viability, IC10 for cytotoxicity. And they're here plotted on a negative logarithmic scale, so which means that no, they're only plotted on a logarithmic scale, which means we have high concentrations, low toxicity for certain chemicals. And you see that for the low potency chemicals, those are the hydrophilic chemicals, they are mainly staying in the water, so there's not a lot of difference. They're not taken up very strongly by the cells. There's not a lot of difference, but as you go to a more potent chemical, there's up to a factor of 10 difference between the measured freely dissolved concentration in the medium and the nominal, that is the added concentration. When you then look at the reported gene endpoint, and that's for the circles are for the AREC32, that's that assay on oxidative stress response that we're also using in PrecisionTox, and the other one is for an endpoint related to the peroxisome proliferator activated receptor. And as you see, as you get, the chemicals are getting more potent. So here we're at the micromolar range. Oh, this is molar concentration. Sorry, I should have said that. These are micromolar concentrations, millimolar concentrations. So when you're at the micromolar concentrations, your freely dissolved concentrations are actually much lower than the nominal concentration. So here it becomes very important that you actually measure the exposure concentrations. So what can reduce, and these are all stable chemicals. So these examples are for stable chemicals. So what can reduce the chemical exposure in cell assays? So when you add a chemical to the exposure medium, and this is pink because the exposure media have a phenolate inside, so it's really pink. So all of that pink I had before was also real photos. So what happens? It's in that medium, you have between 0.5 and 10% protein and lipid-rich fetal bovine serum normally. You really need that for the cells to grow well. And then you also encounter volatilization. Chemicals can bind to the plastic of the well plates. You have to use some a polystyrene well plates, otherwise the cells will not stick to it. They won't stick to glass. When you work with 2D cultures and want to do imaging, have to stick, and so you have to live with the fact that you're working with polystyrene wells. And then you have a certain kinetic and uptake, so your assay will usually last for 24 hours. So the question is, will the chemical reach in that time the cell? And then you also might have metabolism. So here comes the first quiz. What do you think, which loss process is most important in your opinion? Is it volatilization? Is it degradation? Is it binding to medium components? Is it binding to the plastic well plate? Or it depends on the chemical? Or it depends on the chemical and the bioassay setup? Or you just can't tell because I was talking too fast.
Okay, thank you. So the first quiz has been launched. We have for the moment 44% of participants that answer the quiz. So feel free to continue to reply. Okay, so slowly people are answering the first quiz. Okay, so we are now at half of the participants. We will stay 30 seconds more. Okay, so let's 10 other seconds. Okay, so we can close it now. So we have half of the participants that answer the first quiz. Let's show the results.
Yeah, I think I can stop my talk now because most of you said it depends on the chemical and the bioassay setup, and that's what I will show you in the next next few moments. Thank you very much for actively participating.
Thank you, everyone.
So I think we can carry on. Now, how do I get rid of the quiz, which is now in the middle of my screen?
Is that okay now, Beat? Yes?
No, I still see it.
You still see it?
So now, what do I do now? Now I can't see my talk anymore, but never mind. But I can still see did I go out of the?
You went out of your presentation. Can
you
close the
because
little...
I want to get rid of the poll.
Can you close the little poll window with the close button?
Do you manage, Valentin? Sorry.
Yes, I close it on my on screen. Beat, you can close it also by clicking on the cross.
Yeah, it's fine.
So now I'm back in the.
All good
for this.
Okay, so. Okay, so the uptake is indeed depending on the medium composition, so on the cell assay, as you said, and it's depending also on the chemical. And with respect to the chemical, on the chemical hydrophobicity and its speciation. And so this is a bit of a complex slide. So you have the chemical fraction in the cell, and it's actually pretty small. It's less than 12% because the chemical is everywhere in the system, not necessarily in the cells. And this is in a Tox21 setup, 0.5% FPS in 6 microliter of medium on 1536 well plates, 1536 well plates. This is another example where you have a system like the opposite. That's none of the ones we're using. That's just the other side, the opposite 96 well plate, a lot of FPS in 120 microliter medium, and then we have less than 3% overall hydrophobicity. I used here not KOW, octane water partitioning, but a measure that we call liposome water partitioning at pH 7.4. So it accommodates also charged chemicals. A bit too complicated, too long to explain you now. But basically, it's also a measure of the hydrophobicity. And what you can see is that a higher medium FPS leads to a lower fraction of the chemical in the cell, but everything is much more stable across the many different chemicals. When you then look at the kinetics of uptake, and this is with some fluorescent dyes, it's with benzopyrene and the positive derivative thereof and the negatively charged derivative thereof, and it's time after dosing, 24 hours. Then you see that the benzoapyrene is pretty fast. It's taken up after pretty much 2 hours. While when you have the chemicals that are charged positively or negatively charged, they take a little more time. So you have to take, but they will be in more or less in steady state by the time you end your experiment. And when we look at BAP, when you look at BAP uptake as a function of FBS concentration, then it's fast. It's 40 minutes when you have a lot of FBS, and it's very slow, or not very slow, but it's a little slower when still under 4 hours when you have 0.5% FBS. So basically, you have facilitated transport by those colloids that can also be predicted. So it's we find what we had, what would be predicted, and so you can kind of adjust the time to uptake by changing the cell conditions. That also means you have to keep your conditions always constant that you get comparable results. Then sorption to polystyrene plate is relatively low for cell assays. And the reason is that the diffusion coefficients in polystyrene are very small. So they are the partition constants are very high, but the diffusion is very small. Otherwise, all your chemical would be sucked up by the plastic because the volume of the plastic is really high. But there are only a few micrometers where the chemical goes into the polystyrene. So basically, we almost have something like a surface adsorption, but in true, it is an absorption process, a partitioning process. And that's why we can model it quite well. And when you look on the right side, you have a mammalian cell assay, this time calculated for 10% FBS in a 384 well plate. So that's, for instance, the AREC32 test. Then for a chemical up to a KOW of 8, we have less than 6% loss of the chemical, and above KOW 8, very hydrophobic chemicals, we're outside the model calibration range. Then we have a fish embryo test, and that's much more complex. So here's one fish embryo in 24 well plate, and then already at a KOW of 4, you start to lose more than 50% of your chemical by binding to the plastic. Just because you have nothing to retain it, you don't have the medium to stabilize it and keep the freely dissolved concentration constant. So that's also one of the reasons why we're talking right now in work package 2 to limit the 250 chemicals to chemicals around a KOW of log KOW of 4 to 5. You can then use mass balance model to estimate the exposure because we now know all the processes, so we have really worked on them over the last years. So Louise Henneberg, Fabian Fischer were the people most involved, but many others, even Julia, who's now with PrecisionTox, participated in part of that work. And we can set up models where when we have the partition constant between the different phases, we can actually set up a mass balance. I'm not going to run you through the equations, just believe me, they're simple and they're correct. And with these equations, you can then predict for which chemicals you will incur loss processes that you then can decide if they're acceptable or not. Here again, 96 well plate, 200 microliter medium, 10% FBS, 22,000 cells. Now we bring the cells into the game. 1536 well plate, 6 microliter, 0.5 FBS. And you see chemicals like naphthalene, they're just lost. I mean, we know they're volatile chemicals, but you wouldn't really expect the pH to be lost. So, well, it's not really a pH, it's just two cycles. But you have to be mindful. So you start to at a Henry constant of about 4, a little bit more retained. When it's also hydrophobic, you start to lose chemicals out of your cell assays. And then for most of the other, for those chemicals in this 96 well plate setup, polystyrene is pretty much, binding is pretty much negligible. And you don't have an appreciable amount in the cells, and pretty much everything is stabilized by the medium. That's not the case for 1,536 well plates, where you incur many more losses and you have a much more uneven distribution to the cells. So with a high medium FBS, you have a large reservoir of chemicals that you can push into the cells. So the exposure concentrations are more constant, even though you only have a tiny fraction. So it's really a difference between concentration and mass balance. And so what we termed this was called was zero-mediated passive dosing, because you can really use the medium and adjust the medium to deliver the chemical to your cell. So if you have a hydrophobic, difficult chemical, you push up the medium composition. Overall, it looks a little bit less sensitive, but the true sensitivity is the same. And then you can get a much more stable conditions. And the other way around, when you have a low medium FPS or another protein as stabilizer, you have rather uncertain exposure conditions, hard to keep them constant and reproducible and repeatable. So you can actually control exposure by adjusting the medium. So you can do that the other way around. We have a little paper that says how to improve the dosing of chemicals in high throughput assays, and those are kind of optimum conditions. So when you have 384 well plates, 40 microliter, about 5,000 cells, you should have 5% or more of FPS to stabilize your system really well. And you can use that to improve your dosing. But you can also use this to aid your quantitative in vitro to in vivo extrapolation, because you know where your chemical is when you do this. Next step is metabolism that we have to keep into account. We have to keep in mind, and here we have two different types of approaches, an extracellular and intracellular approach. And what we are looking into in PrecisionTox is the metabolism performed by the cell lines. And the 3 cell lines we've chosen that I introduced already, we've chosen for PrecisionTox, will initially be reported that they have no metabolic activity, but they can actually be activated by the chemicals. And so when you have the 3 cell lines, ARE32, oxidative stress response, ARE blah, so you for the ARE blah cell line is based on a HepG2 cell line, so it already has constitutively expressed cytochrome P1A1, but for the ARE32, which had been rightly reported not to be metabolically active, it can be really well induced by chemicals like benzoapyrene. So we don't know yet how this is going to look for the PrecisionTox chemicals, but this is the type of techniques we'll be using. And then as kind of a metabolism control, we would have the GR+ cell line that we're also using that's based on a HEC293 cell line. And we could actually also set up a little model that has those components. So we have an uptake into the cell. We can measure that with fluorescence, time-resolved fluorescence measurements. The BAP will metabolize to fluorescence and non-fluorescence metabolites and be excreted again, and we can measure that in the cells. So at first we look at GR blah that has no metabolic activity, and with this we can fit the uptake rate constants from medium to cell, and the excretion rate constants from cell to medium, being here in black the signal for the cells, and in red the fluorescence signal for the medium. And this we assume is constant for all cell lines, the uptake and elimination rate constants. So the additional what we have to fit when we see these curves that clearly say, oh well, you're taking up into the cell, but we are also already decreasing it again, and we're depleting the medium. And with this, we can fit the metabolic rate constants for the two systems, and they're fairly similar. So that's the way of things we can do and look a little bit closer into the cell line and better understand what's really going on inside. The next thing that helps us is baseline toxicity. Baseline toxicity is the minimum toxicity of every compound, and we've been using that as an anchor for the evaluation of specific effect. This has been a very successful concept for the evaluation of aquatic toxicity, as early, or actually 1899, Meyer and Overton showed in Zurich, showed the tadpole narcosis, kind of the baseline toxicity concept. Then it became very popular with the work of Hans Könemann in the 80s, was used highly in the 90s by people like Lynn McCarthy, Don McKay, and has then really been an established concept that has already been used in a big Pelston workshop in the 2000s. And we can use this also for our approach here. So baseline toxicity is in principle, as I said, the minimal toxicity of every compound. It's related to the membrane function, the membrane that functions as a barrier, where energy transduction takes place in the mitochondria, and which is a matrix for protein, very simplified. So chemicals can make pretty much holes that lead to decay, can integrate in the cells, that lead to holes that lead to decay of the potential, so that no ATP can be synthesized, and there's decreased membrane activity. And so that has been shown for all those aquatic animals that I mentioned. So now the question is that also applicable for in vitro cell assays? So Valentin, if you can put up the quiz. So the question is baseline toxicity applicable for in vitro cell assays? And the answers are yes, absolutely. Or now I can't read it anymore. I know here. No way. This was derived for tadpoles and fish and other critters in water. And the next one would be maybe for cells, but not for Drosophila. That's our running joke in Work Package 2.
So again, the quiz has been launched, so participants feel free to answer the question. So we keep it open for a minute. Okay, so we are already 20 participants that respond in the quiz. It can do better than the first quiz. Come on. Okay.
Who is the one? That's Brian.
Okay, we can keep it more 15 seconds if we are other participants that want.
I think we're at 96%. This is not going to change anymore. That
Okay.
one loner who thinks it's not applicable for Drosophila, that's my target, to show that in PrecisionTox. Anyway, so the baseline toxicity concept is very well applicable for reporter gene assays. We have measured baseline toxicity Q cells where we looked at the, again, hydrophobicity, membrane affinity as a function of cytotoxicity here, negative logarithm of that IC10, and they pretty much lay on top of each other, these different Q cells, quantity, structure, activity relationships, and they don't differ much between each other. Why they differ is really that makeup of the bioassay that I had just shown you with the different medium compositions and everything. Okay, and so again, you can model that. So you can go from external to internal concentration and model the critical membrane burden, and you get a fairly constant. This is experimental data transformed into critical membrane burdens for cytotoxicity and the different cell lines, and they don't differ between the different cell lines, and they're around, the mean is 6 to 9 millimole with a confidence interval between 50 and 90. And we can take this model and can take it a little further. First of all, we can compare that with algae, daphnia, and fish, and hopefully soon Drosophila. And we will find out, so the endpoint here is internal effect concentration for 50% of the effect. So growth rate inhibition for algae, immobilization for daphnia, lethality for fish. But you see they're all in the same range. So the same concentrations in the membrane disturb the animals to the same degree as the cells. I mean, that's kind of a no-brainer because animals are made up of cells. So you were all agreeing on that. We can actually expand that, and I think for sake of time, I'm going to jump over that, how we expanded this model, because it's also a bit complicated. So I go one step further. But the paper is there, so I can send it to you if you're interested, how we expanded the baseline toxicity QSAR to both ends of hydrophobicity. But how can we then interpret the data? So we can compare an experimental data point. That's also what we're going to do in PrecisionTox. Compare an experimental data point to the baseline toxicity. That's called toxic ratio. That's a very common denominator in aquatic toxicology. And we can also calculate the specificity ratio. On the one hand, how far we are away with the experimental QSAR from the baseline. So that's our specificity. And how far we are away from the experimental cytotoxicity. So that's the selectivity of the chemical for that particular assay. So if you want to make a drug, you want to have a high SR cytotoxicity. If you want to make a chemical that's in a consumer product, you want to have a very low SR baseline and very low TR. So and very low specificity. So it's really something that allows you to weigh the specific effect of the chemical that is triggered, and then for every endpoint, for every reported gene assay, the SR and TR will of course be different. And so we analyzed the Tox21 database for those assays where we also had experimental data. So all of that is we showed first with our own experimental data, and then we applied it to the whole database. And we showed that about 50%, a little more than 50%, a little less than 50% of the chemics in Tox21 were toxic. We had more than a toxic ratio of 10 with respect to baseline toxicity. So that's cytotoxicity, and very few were actually specifically acting. I mean, these are still considered between SR1 and 10, could still be considered as causing that specific effect, but it's not highly significant. It's not highly selective. So we have a smaller number of chemicals that in some cases have exceedingly high specificity. So when you have here 10 to the 6, so they are 10 to the 6 times more potent than their cytotoxicity. So they are the guys to watch out for. We apply these HTS cell assays for many different purposes. So you can use them for profiling single chemicals, treatment efficacy of natural treatment systems, for surveillance and compliance monitoring, for a lot of different things. And you can apply them in many different formats. So I talk mainly about cell-based assays, but of course there's many other small animals, bacteria, etc. you can run in those formats. What's really important is a good quality control, quality assurance with respect to accuracy and precision. I'm not going to go into that detail, but I want to just bump up the next quiz. So I've thrown pretty much everything into my in vitro bioassays. So what have I not added yet? Drinking water, microplastic, turtle blood, beluga whale blubber, my own earwax, designed mixtures, urine.
Yep, so the poll has been launched. Thank you, Beat. So participants, feel free, as we did for the 2 previous ones, to answer. So the question, we have different possibilities. Okay. Different propositions, so we will make sure that everyone has the time to read it and answer. There is kind of a battle between her own earwax and dust for the moment. Okay. Still have a few seconds, so please feel free to continue answering the question.
May I?
Yeah.
So, do you know anything about the filters in the size code? Okay. All right, never mind then.
What happened?
Yeah.
Okay, I think we can end the poll. Of course, you can test after extraction, you can test your earwax. That's actually a very good non-invasive measure. When I tried that for the first time, I didn't hear for a while, so you have to do it a bit more professionally, but that's of course, that's absolutely possible. What doesn't make any sense is urine really, because you get the metabolites only, so you don't really get the potency that is of relevance. And everything else after extraction you can throw in. What you wouldn't throw in is microplastic itself, but you would have to extract the chemicals in microplastic, because evidently if you just take a piece of plastic, you'll get a partitioning experiment like you'd get in anything I've shown you, and you just get kind of a passive dosing device that's not very efficient. Okay, so now I have to really rush. I'm actually starting to go over time. That's the quizzes. we apply it for human health risk assessment if we can do quantitative in vitro to in vivo extrapolation. So I don't want to go into details. You can read that up. It's also not our work, but the great work of US EPA. You can apply it for environmental risk assessment. From the chemical perspective, you can use the data, for instance, from Tox21 or your own measured data and come up with predictive models. And that's a little bit, in a way, what we're going to go and do in PrecisionTox, also which is going to go and take it all the way through to the human. And we can use it in environmental monitoring. And here we combine chemical analysis and in vitro bioassays. And so this is an example of testing water quality during rain events. And we have kind of a margin that's safe. That's a so-called effect-based trigger value. And if chemicals, for instance, in untreated wastewater are often above that trigger value, and once you have treated it, and once in surface water, you get below the trigger value. But if you have a rain event, a lot is happening. When you have a rain event, you get crazy mixtures, and every rain event is different, and you can measure these mixtures, and these are actually mixtures modeled, but also redesigned in the concentrations that they were detected and checked for the mixture effects that were all concentration additive. And then we can go one step further and can kind of see these are the chemicals we detect, but they explain only less than 1%, typically between 0.01 and 8% of the effect caused and predicted or even measured by the detected chemicals that we mixed again together. And within those known chemicals, we have many components contributing. We have some that we see all the time. And when we look at those names, I have no time to go through them, but some are very well known, some are quite unexpected. But especially during rain events in that paper that I'm citing here, it was a lot of variability between sites. At one site, every rain event was different. We can also apply these assays in biomonitoring. And so we've extracted brain, liver, blood, and adipose tissue that was post-mortem tissue just for method development with passive sampling with polymers, and submitted them both to GCMS and in vitro bioassays. We find the usual suspects, DDE, PCB 153, increasing with age, but we also find in almost all of the tissues, we find non-persistent compounds. And here again comes in that mixture effect. And so we tested that also in bioassays. And this example is a real hydrocarbon receptor activation. We looked for 100 chemicals, we found 35 chemicals, 5 of them activated the real hydrocarbon receptor. And the more active chemicals we had, the more effect we could explain, but we could only explain up to 1% of the effect. That's only a starting point. We're just moving into that space. I think that's really the future to access the human exposome via these in vitro bioassays. Okay, so with this, I want to come to a conclusion. I hope I was able to show you by a few sketchy examples how cell-based bioassays have a remarkably wide applicability in hazard and risk assessment, as well as for environment and human biomonitoring. All organisms are made up of cells, and so they cannot describe, cell assays cannot describe the intricate interactions on the organism level, but we're pretty good in describing in a very robust and a very repeatable way the initiating and the key events of toxicity. Hopefully, the work on the cellular level may be of use for PrecisionTox, and I'm very curious of what you think about it. With this, I would like to end, and I would thank especially work package 2, who I only know as Tiles, but always happy, smiling Tiles. I would also acknowledge our research unit chemicals and the environment at your set. You're just standing in front of a DHL truck. So that's by chance that we get this nice logo. And this is my department, cell toxicology. who have all done that stuff that I showed you. Of course, there's more people that you have said that you'll meet over the time. Especially, I would like to point out here Stefan Scholz, who has been the leading driver within PrecisionTox of the UZ team. Thanks very much.
Many thanks, Professor BEATE, for this really interesting talk. So we are right on time, and I've already few questions in the chat. As a reminder for everyone, so we have now half an hour to take your question. So you can write it in the chat, or if you want, you can also raise your hand by using the reaction button on the top of your screen, and Agatha, Jeanne, and I will be happy to give you the floor and ask your question to BEATE. So first, there is a question from François Busquet. So François, you can turn on your video and turn on your mic. So the question is, BEATE, are you considering to investigate the fetal calf serum free database and see its impacts on chemical stability?
Yes, thanks for that question. Yes, we are moving towards totally animal-free test systems. So we are looking into chemical defined medium. So we've tried a few things. It's really hard to get cells then to still grow and not die immediately when they see a chemical too. But we have an NC3R funded project that was just finished and will continue for another couple of years, where we are targeting fully animal-free media. But it's not easy, and we do that together with much stronger partners. So again, we will be responsible more for the what does it mean for the freely dissolved concentration, what does it know for the metabolism and everything. So we are not the cracks in the media itself, but in the characterization.
Okay, many thanks, BEATE. François had a thumb up. So we can go to the next question. So I see that a hand raised of Stefan.
So that's Stefan, who pretends to be in disguise here. I have to introduce you. Valentin is new, so how can he know who you are, Stefan?
I don't know. That's strange. I only realized that I have this strange acronym. I wasn't aware of this until you said. That's crazy. Yeah, finally I managed to get into this talk. It was a bit of a challenge. So my question is actually related to what François asked before, and it's related to these proteins which we find in the cell culture medium, but then if in this serum-free media, there are probably other proteins or whatever included. So there's kind of two questions. First, would these ingredients in serum-free cell culture media also stabilize the concentration? And would you actually recommend to add something like this, proteins also, to the non-cellular models like Xenopus or Daphnia to stabilize concentrations? Or do you think that's a bad idea?
I think that's a bad idea. I think that's a tough one. I mean, certainly for the cells, I think the important step is to go to chemically defined media. And then you can add some more benign protein, not a fetal protein, to kind of stabilize the medium. And you really need that for the cell assays, because in the 40 microliter, I mean, I was enthusiastically talking about the measurement of the free concentrations, but it's actually still pretty hard. So it's nothing we can do on a routine basis. And we're not going to do that for the 250 compounds. That's just impossible. It's really challenging. So you would be developing a defined medium that has some sort of, you can almost say it like a buffering capacity. Yeah, so, and then for the other assays, no, I wouldn't really start to fiddle around with colloids of unknown nature that could disturb. Here, once you go to more hydrophobic chemicals, for instance, fish embryo toxicity assay, passive dosing devices are pretty good that you can use. So you have to go another way. I think it was also in most of the in most of the other assays, you have much larger volumes. So testing the chemical concentrations is not a big problem. And because the medium actually doesn't have a lot of growth factors and proteins and all of that, it's also easier to measure it. I mean, you can't just standardize measure. You have to get either precipitate the proteins. So for us, really getting that SPEMI method up and running for neutral and charged chemicals, but you still need a different volume and different SPEMI for each type of chemicals, was actually something we had to do in response to the fact that you can't, like in a flow-through test in a fish, also that you can continuously monitor the aqueous concentration because you have liters over liters of medium, and even in the fish embryo you can just take a bit out. It's pure water, you can inject it directly. So I think this, I wouldn't go that way from the cells to the other organisms. But some of that, when people say, oh, cells are not very sensitive, they are as sensitive as an organism, just their nominal concentration might be higher because of that sorptive capacity of the medium.
Okay, many thanks. Stefan, you say you had maybe another question, or is it okay for you? It's good. Okay, great. So we can go through another question from Luigi Margiotta. Please, Luigi, feel free to turn on your mic and your video. You can ask directly the question to Professor Beat.
Yeah, the fantastic talk. Thank you very much. I did learn a lot today. I have a question about inter-individual variability in vivo. Considering the high variability that we sometimes see in the individuals in a population, how do you think we should address this from an in vitro testing perspective? Do you think that modeling is the way to go, as for example, we do for some cardiotoxicity testing with stem cells-derived cardiomyocytes, or do you think that we need more in vivo toxicity data to generate more accurate in vitro to in vivo extrapolation? Well, I think the variability is really also very much coming from the in vivo receptor. And so there's a lot of high throughput toxicogenic modeling that's involved there to get from, like, once you have the plasma concentrations scaled up from in vitro to in vivo, what that really means in terms of dose for the humans. And I think that would be the way to go. I wouldn't really start going, I mean, personally, I wouldn't really start doing a lot of more animal testing, but use all those advances in high throughput toxicokinetics, also from population data, and rather read across from other chemicals than to do a lot. I mean, I'm really an experimental person, but I think that's the wrong place where you should do a lot more experiments. Thank you.
Thank you very much, Luigi and Beat. So we had a question in the a remark in the chat box that Anna Riquel were asking if you can maybe propose the paper that was about, if I misunderstood, the cytotoxicity.
Yes, and I would be great if you send me an email, then I can send it back to you, because okay. I'm not sure right now.
Okay. Okay, if you have any other question, feel free to raise your hand and write it in the chat. We have enough time now. Okay, Anna Riquel is replying that she will send you, we'll keep in touch. We have another question from the chat. Thank you, Georgia. The question is, are you considering human iPSC derived model for chemical toxicity assessment?
We would like to, but at the moment our facilities would not allow this. So at the moment we're really in these really high throughput, very robust systems that are also the same when you test them today and you test them in 5 years and you test them in 10 years. So we do a lot on water quality assessment, and we're going into the human biomonitoring. So at the moment, we're really into those really robust tools. I think these more advanced models would be beautiful, but to get them to a stage where you can run them
in such a high throughput, it would still take some time. And unfortunately, we don't really have the facilities yet.
Okay, so maybe it answers also the question that has been asked by Francois, why can we use iPSC? Okay, another question or maybe remark from Stefan about how to discuss the Drosophila baseline toxicity.
That's a good one.
I wasn't actually the one that had this minority vote. Just say that. Yeah, I briefly want to explain what I mean here. But I think it's really great that we have this Drosophila model in and can compare it also to the others. But I think it is really challenging because the uptake of chemicals is totally different because they are fed by the chemicals, and we learned even that sometimes they avoid feeding when there is some chemical included. I'm very skeptic whether we can demonstrate this, but I'm absolutely curious to see how compounds that are identified as reacting mostly at baseline in the other organisms, how they show in Drosophila. I think that will be really interesting.
I still believe, I mean, if we can extract and measure, we could still establish the total body burden. And then when we have a model about the components of Drosophila, I mean, it's protein, lipids, and some non-sorptive residue, which I would like ketene or so. I would assume that whatever they have would be not very much sorptive, at least for the more hydrophobic chemicals. Then we could at least get critical internal concentrations. If we can model them back to the dose, might be a little bit complex, but then look at humans, you know, I mean, we breathe, we touch things, we eat things, and it's fairly complicated. Still, we can get a measure from the external exposure to the internal exposure. We have amazing models. And so why not being able to do that for Drosophila too? In the first step, I would definitely start measuring to have that kind of real anchor, an experimental anchor, like we did for all the other test systems over the many decades of work on baseline toxicity, because it's really nice anchor. And then this distance, the toxic ratio and the specificity and selectivity ratio, then it would be really interesting how much they differ in these organisms that are more susceptible to insecticides, for instance, etc. So I think then we would just have something much nicer to compare with. And I think it's almost imperative to do this, because otherwise we just get numbers that we can statistically compare and see if they align or if they associate somehow. But I think to get the critical internal concentrations, and then from that model, the internal membrane concentrations, I think that should be a feasible way of doing it.
Yeah, I fully agree. But then we should kind of discuss in the project that we're trying to get this analytical data for Drosophila, right?
Yep.
Thank you, Stefan. Okay, so for the moment there is no hand raised. So maybe last call for question. If anybody wants to ask a question to BEATE, please feel free to raise your hand or write it in the chat. Okay, so I think we can close this second session. Thank you very much for taking time to attend this webinar, and a big thanks again to Professor BEATE ESCHER for this very nice presentation. For all of you, stay in touch and visit our website of PrecisionTox. So I put it right now, the link in the chart. And we will also provide you with a survey link. It is always important for us to have your feedback about the organization, the content of this second edition of the Café Scientifique. We keep you in touch about the next one, the date and the topic. So you can now have access to the PrecisionTox website in the chat. So, okay, thank you very much. Beat, if you want to say something, maybe.
Yeah, well, thank you for thank everybody for your interest, and feel free to contact me because I know I was really racing through everything. And I'd also really like to thank Agatha, Valentin, and Chan for organizing this really, really well. Thank you.
Thank you very much, Beat.
Bye-bye.
Okay. Bye.
Bye-bye.
Thank you.
Cheers.